In the first discussion, we gave you a little bit of background and hopefully some foundation for moving on to more complex topics. We're really going to jump in. If you've already done the reading for this lecture, then you'll realize that it's getting kind of serious, right? It's a little bit complicated. Don't stress out too much about that and don't freak out. This reading, parts of it are pretty technical. Not all readings will be like that, but some will. This is an advanced class, and so we're going to tackle some advanced topics. If you have not had an evolution class before, it's really going to be a stretch for you. That's really one of the primary prerequisites and some understanding for this class. You can still pass the class and even do well in the class, but you may need to reach out to me as you're doing it. All right, so we're going to start this lecture from basically kind of the same premise we ended up with in the last one and then move on to more complex topics. So I've got 4 DNA sequences of the same gene from 4 different insect species. We got a fruit fly, a house fly, a mosquito, and then an apple maggot fly. These are all dipterans, all part of the same order of insects, and so they're fairly closely related to one another. And when we look at the DNA sequence for this gene, you notice that there's lots of variability, lots of differences. These represent historical mutations that are now present and create the diversity and some of the differences between these species. And as we talked about, we're going to, and you'll be using this tool in the future, but we can look at the DNA sequence, we can translate it into the protein sequence, and this is something that is quite common, especially for really important genes. You'll notice that when we look at the DNA sequence, there's lots of diversity and variety. There's even an insert in one of these species, an insertion-deletion event, which we talked about, right, where we're missing 3 base pairs. But lots of point mutations, lots of substitutions. And when we translate it into the protein, yet we have very few. In fact, I don't even see any differences until we get down here to the end where there was that insertion-deletion event. But other than that, at the protein level, it's all the same. So the vast majority of these mutations are what we would call silent mutations, meaning that they're real. We can measure them at the DNA level, but when we translate this gene into a final protein product, there's no variety and no diversity at all, and that's why we call them silent. So these would have a genotype difference, but no impact on the phenotype until we get, again, to these set of mutations down here. Okay? So again, this specific example is not critical, but it shows you something that we see very, very frequently, and we'll be dealing with this a little bit more. So as biologists, as molecular biologists who are studying this sort of thing, we want to be able to analyze this. We want to be able to use this data to help us understand maybe the relationships of these organisms, maybe the way the gene is evolving and how that evolution of that gene might impact or not impact the phenotype of those organisms. So again, just kind of base, and that's where we're going to be going from. And we'll come back to a DNA alignment here in a little bit, but we'll use a little bit more of a simple one, and we'll simulate and talk about some of the aspects. Now, I also want to make it clear, because we're going to make this connection, we're going to talk about populations later in the class, some of the underlying processes that are going into creating all of this genetic diversity that we see in a gene. So to do this, I'm going to give you a very simple diagram here. Well, let's explain. Maybe it's a little bit less simple than I might originally have. If you're experienced and look at it, it looks quite simple, but we need to give you some background so you understand what you're looking at. In this diagram, the big tree, this big, bold, fat outline represents the species history, the phylogeny, right? So we have species A that broke off anciently from a common ancestor. We have species B that broke off a little bit later from the common ancestors of B and C. And so we have relationships among these 3 species, and that's what the big tree represents. The smaller lines inside this represent alleles. So we have each dot representing an allele with a line connecting it to its offspring, in which, excuse me, its offspring that inherited that allele. Okay, and so this is obviously a very, very small population. This is not realistic for real live populations, which might have tens of thousands or millions of individuals, but it's easier to visualize. And some alleles get passed on. In this case, we have this allele that was passed on twice just by chance, and the other allele in that individual that was never passed on despite this individual reproducing twice. Some alleles are passed on a lot. Other alleles never get passed on because that individual maybe dies out before it can reproduce or doesn't reproduce. Or maybe it's just by chance. An individual reproduces a single time, only one allele can get passed on. Okay, so this shows you both the species history, and then inside the bigger phylogeny, you see a little bit of the allele history, or the gene history, if you want to put it that way. Okay, so genes get passed on. Now, I've also put one more important process here going on. Notice that here I've got a red line there. That represents a mutation. So when this allele was copied and passed on to its offspring, a mistake occurred. And this could be just one single substitution. We were looking at DNA alignment. This would be just one of those many changes that we saw historically in those species. Now notice when that mutation first occurs, it's simply one of two different genetic variants in this descendant population. So here for this gene, there's no diversity at all. We represent that with the black colors. If I was to look at the DNA sequence of this allele, ACTG, it would be identical for all 8 of these alleles present in the population. Homogeneous, every individual is a homozygote. There's no genetic diversity at all. But we have a mutation, and suddenly genetic diversity is introduced. And originally, as I said, this is just one allele, one variant among many, many. But if this variant provides an advantage, then it will be passed on at a higher frequency because those individuals do better. They reproduce at a higher rate. They're overrepresented in the next generation. And that's called natural selection. So this is a positive selection for this gene, a form of natural selection. And eventually, if it's a positive enough, we're going to end up with complete fixation of this allele. So now every individual takes a little longer for population C. And notice that when this population broke up, the ancestral population, right, which is represented right here, that when that population broke up, there was still variety and diversity, and that was lost very quickly in species B, and then a little less quickly, but still happened in species C. And so eventually, species C and species B all carried, they're homozygous again, they all carry this new variant that is distinct and different from species A. So for phylogenetics, that's great. That becomes a marker that helps us determine and distinguish the difference between species B and species C. Now, there are many complex processes that we'll be talking about in later chapters that can make it a little bit harder to determine whether or not this marker is really a good marker for those relationships. But that's something later. For now, I just kind of want you to get in your mind and your head this idea of going from a population and then a process of fixation through natural selection. It can also be just simply fixation through random chance, through genetic drift. But eventually, fixation of alleles leads to distinct markers that unite certain groups of species and distinguish them from other more distantly related species. And so that's the underlying process. And when we get to our section on population genetics, we're going to look at this process within the population. But for now, everything we talk about is going to be just looking at differences between species and what these complex underlying processes lead to. So with that in mind, let's then look at DNA sequence, the result of this. So we're going to switch from this more complex view, right, where this is real DNA sequence that I downloaded from the database, and you'll learn how to do that and do that in some of the worksheets. But let's look at a very more, it's artificial, but at a simpler view that will help us to understand this process. Now, when I teach this class in in-person format rather than online, I do this exercise where I actually pass out a sheet of paper to individuals with the sequence. I have them simulate mutations, and then we gather them all back, okay? But we're going to do this. It's probably not as helpful, but hopefully you can kind of see what's going on and wrap your mind around this. So to do this, I am going to create a brand new alignment. So let's get rid of some of these. In fact, let's get rid of all of them. I'm going to add a brand new sequence here. We'll get rid of the original DNA sequences. And let's cut and paste in. I think I can just do this rather simply. Let's cut and paste in this DNA sequence. I think I can just copy the text and paste it in here as if it was real DNA sequence. Because of the spaces, it's not going to like it. So I'll just type it in. All right, so we'll make sure we get it exactly the same as there. So let's say we start with a sequence that looks like this. And I've just randomly picked a DNA sequence. It could be any sequence, really, okay? So this is going to be our very first sequence. Just so we don't get confused, let's call it sequence 1. All right, now, as this DNA sequence gets passed down, most of the time it's going to be passed down exactly the same. There are going to be no mutations at all. But every once in a while, we will get a brand new mutation, right? So if that occurs and we pass it down, let's say we have a species that splits off and it gets passed down, but when it does, there's a new mutation that occurs. So our sequence number 2 is going to look nearly identical, but maybe we have one mutation. So let's just put in a single mutation. I just randomly picked one. Now, when I do this in the class, what I do is that mutation gets, or that DNA sequence gets passed on to 2 people. And those 2 people each make a random mutation. So one person might put a mutation there, and then the other person may very well do the same thing, but maybe they choose to put the mutation somewhere else. Oops. So let's go back to the original sequence, and maybe they choose to make that's where their mutation is, right? So we now have 3 sequences, and they all carry a different DNA signature, right? And so if we were to look at this and figure the mutation rate, we could compare DNA sequences, and we would compare sequence 2 to sequence 1 and say, oh, look, there's one difference. We would compare sequence 3 to sequence 1 and also see one difference. So this is a nice, even mutation rate. Everybody's the same genetic distance from one another. And so we are going to define this idea of a genetic distance. Okay, and a genetic distance is simply how many mutations have occurred to separate different organisms. So to calculate a genetic distance, it's very easy. All you do is count up how long the sequence is. In this case, we have 9 nucleotides. Count up how many of those nucleotide positions are different. And we could do that as a frequency, is usually the way genetic distances are, where 0 represents that there's no distance at all. So if I put in a 4th sequence here, AT, we'll make this the same as the original one. Let's say we have a different species, but when it was passed on, there were no mutations at all. So the genetic distance between sequence 1 and sequence 4 is 0, right? Because there are no differences at all. The genetic distance between sequence 1 and sequence 2, there's 1 out of 9 differences. So it'd be like, I don't know, what is that, like 12, 13%? I'm not good with math. So whatever, 8 out of 9, right? And 8 out of 9, whatever percentage that is, and we would make that. Well, let's do it here. Rather than just, I'm going to need a calculator. So 8 divided by 9 is 0.88. So the genetic distance, the differences between these would be like 12.22222. So a little bit more than 0.12 or a 12% genetic distance. And so the easiest way mathematically for us to represent that is a number between 0 and 1. If you want to think of it as 0% to 100%, that's fine too. But we're going to represent it as a frequency between 0 and 1. So the genetic distance between sequence 1 and sequence 2 would be 0.122222. Okay, so a little bit more than 12%. Now, if we then imagine that sequence number 2, right? So let's get rid of these other ones, just to, at least sequence 4, just to simplify it a little bit. Let's say that sequence 2 goes on and it passes on that genetic material to a new species. And when it does this, so I'm going to put it down at the bottom here, there's a second mutation. So we'll write it out here exactly as it was before, But let's say that instead of exactly the same, there's a new mutation. And we'll just pick a random spot. Let's say that one turns into a T. Okay? And we're going to use only substitutions. I could have an insertion or a deletion, but that makes it more complex. So the genetic distance between sequence 2 and sequence 4 is still the same. It would be just a little over 0.12 because I only have one difference. But when I compare sequence 4 and sequence 1, there are now two differences. So we'd be close to 25% or 0.25 genetic distance between sequence 4 and sequence 1. And so as these sequences accumulate, we end up with more and more and more differences. Okay? And so that's the process that we're looking at. And hopefully most of that makes sense in your head, and you've got this idea, okay, DNA sequences being passed down, genetic distances are accumulating and accumulating and accumulating. But I want to give you just one example, and then we'll look at the slide and look at more possible examples, and we won't refer back to these sequences. So we know that sequence 2, because of the way we did the simulation, that sequence 2, right, was the ancestor, if you will, of sequence 4, and they only have one genetic distance between them. However, sometimes, and this is going to happen fairly soon in this small sequence, sometimes new things happen. So let's say that sequence 4, passes on its DNA to a new species. Sequence 4 species is still there, it's still the same, but it passes on to a new species and there's a new mutation. So now we're going to have sequence 5. And just by chance, this mutation occurs at the same position where we had an earlier mutation in sequence number 3. And let's say that, again, this is not a huge possibility, but it could happen that it's the same mutation that occurs earlier there. So I'm going to change it so it looks just like sequence 4, except we're going to change this, I think it's at the 7th position here. I guess we had 12. I said 9 earlier. Did I say 9? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Oh, maybe you caught me on that. I calculated the genetic distance wrong because I didn't count how many base pairs there were. So it was actually, my math should have been like this. And I won't ask you to calculate genetic distances, but if you caught me, good on you. If not, this is the way it should have been. We should have actually had 11 divided by 12, which is a 91.6% similarity, or basically like an, what would that be, an 8.4% difference, right? 0.84. So instead of a genetic distance of 0.12, it's really 0.084. But again, I don't care that you're able to calculate those, but if that's making sense in your head, great. If not, just put a little pin in it and we'll worry about that at a much later date. Okay? So now notice, let's come back to this. Notice sequence 5. Sequence 5 is only one difference, 1 out of 12 differences from sequence 4. So it's very closely related. The genetic distance between those is about 8%, a little bit more than 0.08. Okay? But when we come back and compare it to sequence 3, notice that even though there have been 3 genetic distances, that there have been 4 total mutations, 1 at that position, 1 at that position, 1 at that position, and then because we simulated it, we know that it was actually a 4th mutation right there. But when we compare them, if we don't know the history of these organisms, if we only have the resulting sequence, we only count it as a genetic distance of 2 out of 12. Right? We count that mutation, and when we look at those two, we say, oh, look, there's no change there. Now, in reality, because we know the history of these sequences, we know there was a change at that 7th position in sequence 3, and then there was a change in sequence 5. In fact, let's take a look. I'm going to pull up a little, let me give you a brand new document here that I can write on. Make this a little bit smaller so we can actually compare it. Give me just a minute. So this, if we draw out the history of these sequences as we have simulated them here, this is how we did it. We start with an ancestral sequence, and then species 1 broke off, and we have no mutations along that lineage. So we'll put species 1 here. We then made a mutation for sequence 2, right? And so sequence 2 broke off, and there were no mutations along that lineage. Somewhere along the line, sequence, and this is the one that we had leading to sequence 3. Sorry, my 3 is a little funny there. But along the lineage to sequence 2, we made another one break off that eventually led to our sequence number 4. And then from that one, we also had another mutation that led to sequence number 5. Okay? So because we know this is the history, we could map the mutations on this phylogeny fairly easy. So there are no mutations leading to sequence 1. The ancestral sequence we know was identical. Okay? Sequence 3, which broke off, we know there was a single mutation right here. And so if I compare the difference between sequence 1 and sequence 3, we only have this one difference right there. So that was at position number 7, a ancestral C mutated into an A. So here we have at position number 7, let's just make a little note, and I'm going to have to erase some of these because they'll get a little bit busy. At 7, we know that ancestral sequence, the ancestral sequence was a C. At position number 7, that ancestral sequence mutated into an A, an adenine. Okay? Hopefully that makes some sense to you. And so if there were no other mutations, then all these would be identical and this one would be the same. We really would have no reason to separate these out if there was no other mutation. But that C turned into an A. Okay, so only one mutation, one difference among the 12 between 3 and 1. And we know that somewhere leading historically up to 2, somewhere ancestrally here, that there was another mutation. And so if I compare sequence 2 to sequence 1, that mutation was at position number 1, 2, 3, 4, 5. An ancestral A turned into a G. So we could make a little note, that one there. In fact, I'll draw a little line coming out here. That at that position there, we know that at position number 5, that a ancestral A mutated and turned into a G. We could designate that something like this. And so if we know the history of these organisms, I can mark every single mutation easily on here. Okay. Now, I won't bother to mark the other ones because we did one leading to number 4, but I do want to do this one leading to number 5. Now, notice, because we know the history of these organisms, when I compare species 2 to species 5, let's put these down at the bottom so we can compare them. There are 2 differences, right? So one that occurred somewhere along this lineage, but there's one that occurs up here that makes species 2 different from species 5. So let's mark that one. And that was this one right here. And it was exactly the same as going on right here. At position number 7, a C mutated and changed into an A. Now, because we know the history of this species, we know that that is what we would call a convergent mutation. Hopefully that's a familiar concept to you. A convergent mutation happens in separate lineages, but looks very similar, or in this case, even divergent. So we would maybe even call these parallel mutations as the technical term when they are exactly the same. And so this is a parallel or a convergent mutation that changes the same position to a C to an A. Now, because we know the history of these organisms, because we simulated it out, we know exactly how it occurred. But if all we had were these sequences and we didn't know this intervening history in between here, it's going to be a little bit confusing for us. We're going to look at that position and say, oh, look, 3 may be more closely related to species 5, even though we know, because we did the simulation, we know it's not that closely related. We might put it as more closely related because of this convergent mutation. And so convergence is one of the biological processes that can make inferring histories when we only have the descendant sequences, it can make inferring that a little bit complex. Now, hopefully most of that made sense to you. If it doesn't, that's okay. I just kind of want you to start to get a feel that this can be more complex than you might otherwise think. So let's come back and let's look at some of the other things that can make these mutations more complex. Okay, so one change, that's easy. The descendant species sequences are different. So in this analysis, we're only going to look at one position at the DNA. We're not going to look at an entire sequence. So we have an ancestral sequence where at one of the positions there's an adenine. It gets passed on with no mutations to a descendant. With one mutation here, there's only one difference. That's very easy and simple. Sometimes there could be multiple mutations. Now notice that if I know the history, I say, oh look, there's a genetic distance of 2 here, 2 changes. But in reality, if we look at the descendant species, we would only measure a genetic distance of 1. I'd say, oh look, this species has an A, this species has a T. There's only one change. But in reality, it may be more complex than that. Same thing here. So here we have 2 different changes in different lineages. We have a genetic distance of 1 when we measure it, but the ancestral sequence was actually an A. So genetic distance is actually greater than what we would measure. We'd measure a difference of 1 when we look at the descendant species. Now realize these ancestral species, we don't know for sure what that ancestral species is. We can't go back in time and sequence the DNA of an ancestor 1 million years ago or 1,000 years ago or any time. Those guys are gone. All we have are the descendant species. And so occasionally, if we have a parallel substitution like we say, even though there were a genetic distance of 2, we're only going to measure, well, actually in this case, we're going to measure no genetic distance at all. So I would look at species A and species B and say, oh, look, they both have cytosine. There's no mutations that have occurred. When in reality, it's possible that more than mutations occurred. And this can get more and more and more complex. Okay? We could have back substitutions. We could have multiple convergent mutations. And so even though it's not as likely, we can have many complex mutations leading to a rather simple result. So the take-home message from all this complexity is that our measured distance or our observed distance is always going to be less than or equal to the actual distance. Now, if we, in this case, our observed distance would be 1, and that is equal to the actual distance, which we know based on this history that we've inferred here, that we have constructed, that's the actual genetic distance is 1. Here we would measure the observed distance as 1, right? There's only one difference between these. But the actual genetic number of mutations that occurred, the actual distance is 2. So in this case, our observed difference 1 is less than what the actual distance is. Okay, so just realize that, and the take-home message or the simplified version is, by looking at the descendant sequences in an alignment, we sometimes are going to miss or not be able to observe some of the sequences, some of the history that occurred. Okay, now, we need to make some distinctions between types of mutations that can occur. And this is important because when we measure these rates of genetic distance in real sequences, we observe and notice some real differences. And so we are going to distinguish between a transition and a transversion. Okay? We've already made this distinction between synonymous and non-synonymous. And if you remember back to our original alignment, you remember that when we translated the DNA sequence into the amino acid sequence, that the amino acid sequence was much, much more conserved than the DNA sequence. Okay, and that's because almost all those mutations were synonymous or silent mutations. And there were very few non-synonymous mutations. Okay, so you should know this one already, right? A synonymous mutation is a silent mutation in the coding sequence that leads to no change at all in the amino acid. So it makes a genotype difference, but not a phenotype difference. Hopefully that's clear in your head. If not, go back and review that. In fact, well, let's go back to the original sequence here. And I think I can undo this just very quickly and go all the way back to our original sequence. It's going to take a little bit. Hopefully there's enough memory there. There we go. So lots of mutations here. When we translate it, very few of those mutations actually result in an amino acid change. And so those would be synonymous mutations. Now we do have some non-synonymous mutations here. This position, we'll highlight it, translate it into our protein, or actually back into our DNA. Notice that there was a mutation. There's only a single mutation at that second position in the mosquito, but it resulted in a shift in the amino acid. So that is an example of a non-synonymous mutation. In this one, despite there being two different mutations, two individuals carry a T, one individual carries an A, and one carries a C. There's at least two mutations there. Yet when we go back to the protein, see where it is, here it is, it's highlighted right there. There's no difference at all in the amino acid. So that would be a synonymous mutation. And synonymous mutations are a type of a silent mutation because they don't impact the phenotype. So hopefully if that was not clear in your head, that helps you to understand what we mean when we say synonymous versus non-synonymous. Now, the other type of distinction that we need to make is a transition versus a transversion. So very simply, when a DNA sequence mutates from a purine to the other purine, that is called a transition. So A is a purine, so is G. Review that if you don't remember it from the first lecture. If a G mutates into an A or an A mutates into a G, that's a transition because we just turned a purine, which is a double ringed nucleotide, into another double ringed nucleotide. So although they're not identical, they're fairly similar to one another. If a C mutates into a T, that is also a transition because a pyrimidine, a single ringed nucleic acid, mutates into another nucleic acid. Okay, that's very similar. Purine to a purine or a pyrimidine to a pyrimidine, those are transitions. If, however, a mistake is made and a purine mutates into a pyrimidine, that is called a transversion. So anytime we go up or down, or kitty corner, in this little diagram, it is a transversion. A purine to a pyrimidine to a purine, any of those mutations are transversions. And this is important because there are molecular differences between purines and pyrimidines. And so whether or not a transition is kept by proofreading mechanisms, there are going to be slightly different rates of that because of those molecular differences between those nucleotides. Okay, so we've already answered this a little bit, but let's review. How do we measure genetic distances between two sequences? Well, what we have to do is all we have to work with is our observed differences. And so we could imagine two sequences where we get mutations over time. And in fact, let's do a little bit of a simulation of this. Let's go back to our original DNA sequences, our alignment, and let's simulate this. Okay? Now we're only going to use, instead of multiple ones branching off and multiple phylogenies, making it more and more complex, let's just do two sequences. And let's make them identical to start with. Okay, so I'm going to delete all of these. There we go. Delete. Let's just make a random new sequence. And in fact, to make it simple, this is not biologically realistic, but let's make it all A's. That makes it nice and easy. Okay? So now let's say we have a descendant species, and originally they're identical. So we have species 1, and then descended from that another species that's all identical. And then we get one of these, we get a brand new mutation. Okay, so there's a T. So what's the genetic distance? Well, we just count up however many total. There's only one difference out of all of those, and it would be some small percentage of genetic distance. Now let's make another mutation. It can be in either descendant species. Let's say another mutation occurs in this species and becomes fixed. Okay, now the genetic distance is we have two differences. And so let's make another one. We'll make a guanine instead of a thymine. Now let's make another one. And again, we can randomly do it anywhere we want. And as we do this, as time moves on, as generations move forward, we're going to have more and more differences that accumulate. And so if I came at some point in time and I said, oh, look, there's 1, 2, 3, 4, 5 differences between these species. 5 out of 36. Oh, sorry, no, I made 50. That's nice and easy. 5 out of 50. So this now would be at a 0.1 or a 10% genetic difference. 1 out of every 10 sites is different in these sequences. Okay? And so if we were to graph this on a chart, let's come back here and make a very simple graph. And on this graph, what I'm going to do is I'm going to put on the y-axis percent difference, or let's just say frequency difference between 0 and 1. I'll do that. Oftentimes frequency is denoted by that kind of fancy F. So that's frequency or the differences between organisms. They're going to start out originally at 0. And so over here we're going to put time. Now sorry for my it's hard to write on the computer screen sometimes. At time 0, their genetic distance is going to be 0. They were A all the way across, and both of them genetic distance is 0. Over some amount of time, they're going to become different. And our difference originally was 1 out of 50, so whatever that is. And then over another percentage of time, they're going to get a new mutation, and a new one, and a new one. Now, if the rate of evolution is very, very consistent, we're just going to get a straight line, and over time, they're going to become more and more different from each other. In reality, sometimes there's a little bit more or a little bit less time between mutations, and so probably I would see something that would be more like this, a little bit of spread between when those mutations occur, making them different. And we could even that out and make some sort of best fit line, and that slope represents a substitution rate, right? How often new mutations get fixed in these different populations. And so this slope right there becomes what is known as the substitution rate. I'll just abbreviate here. Okay, hopefully that makes sense to you. And so what we could do is we could come in and we could measure these DNA sequences, and if we had some information about the substitution rate, we could estimate the amount of time, where I know that if I've got that amount of difference, then I have some amount of time that's passed. If I'm measuring a lesser amount of genetic distance between the sequences, I can, again, extrapolate and figure out this lesser amount of time. So this could be very useful if the mutation rate, or rather, the substitution rate is consistent. It becomes a very, very nice way to estimate the amount of time that has diverged since these sequences diverged from one another. However, there's complicating things that occur. So if we have a convergent mutation, so if for many, many thousands of generations there was a guanine at this position, and the ancestral one, what we know, was an adenine, but what if a convergent mutation occurs? So now I measure genetic distance. There's no genetic distance now anymore because these are identical. I would measure the genetic distance as less, when in reality, we know that 1, 2, 3, 4, 5, 6 mutations have occurred. I would only measure it as 4 mutations because at this position, right there at position number 38, they are identical. And so what occurs is as convergent mutations occur, as back mutations occur, let me just give you an example of a back mutation. So imagine instead of that mutating into a G just by random chance, a mutation occurred at this position. Oops, sorry. A mutation occurred at this position. And although we know for many thousands of generations there was a guanine there, just by chance it mutated and mutated back to an adenine. That's called a back mutation. And so even though there are 2 mutations that occurred, we'd measure it as 0 mutations, because when we look at the descendant mutation or the descendant sequences, there are no differences. So because of convergence, because of back mutations, because of multiple mutations occurring at the same site. So here's another example. Let's say at this position, a mutation occurred later in this sequence, and that adenine turned into a guanine. Now we know, because we did the simulation, that there are 2 mutations that occurred. But when we looked at it and compared the sequences, we'd only see 1 difference between them. So instead of 2 mutations, we would count 1. So because of convergence, because of back mutations, because of multiple mutations occurring at the same site and covering up past mutations, we are not going to be able, after a certain amount of time, to accurately estimate the time that has passed based on genetic distance, because some of those mutations get hidden. Okay, so back to our PowerPoint now. And so what's going to occur is this initial slope here, right? This initial slope, using our observed distance, represents the mutation rate. But over time, we have all of these convergent mutations, all of these back mutations, all of these secondary mutations occurring at the same site, covering up historical mutations. And so we end up with what we call a saturation curve. As more and more mutations occur, it becomes less likely that we have a brand new site with a new mutation, because there have been so many mutations that occurred. We're going to have more and more convergent mutations, more and more back mutations, more and more secondary mutations, all covering it up. And so we end up with saturation. And so notice that in the early stages, the amount of genetic distance, base pair differences, is a pretty good estimate of how much time has passed. But as saturation occurs, we have less and less of a way to estimate time. And so the observed difference, because of saturation, eventually reaches a plateau. Now, before it reaches a complete plateau, we could potentially use a correction to help us correct for the number of changes that have occurred. A model of sequence evolution or a model of evolution is how we make that correction. So this is why models of evolution are important. They allow us to correct between the observed distance and the expected distance. Okay? Now, there are differences sometimes between different types of mutations, and this is why we made the distinction earlier between transitions and transversions. Proofreading mechanisms, when those mutations first occur and whether or not they're allowed to stay in the population, the proofreading mechanisms catch transversions much more frequently than they catch transitions. And so transitions tend to reach saturation points much more rapidly than transversions. And we're not going to worry about the mechanism that has to do with the DNA molecule and whether there's a bulge or a pinch in the DNA molecule and how good the proofreading mechanisms are at catching those bulges and pinches. If that doesn't make sense to you, don't worry about it. Not a big deal. All you need to know is that transversions get caught much more frequently by proofreading mechanisms, and so they are removed at a much higher rate than the transitions. And so because of that, transitions reach a saturation point much more rapidly than transversions. So if I want to estimate divergence times based on genetic distances, I could do it for a much longer period of time over using transversions. But if I want to do it over short periods of time, a transition might be a better estimate because there's just more discerning power because more of them have occurred. All right, that wraps up our discussion number 1. It kind of lays the groundwork. We're going to look in discussion number 1.2.2. Our next discussion, we're going to actually start looking at real models of evolution that are used and give you a definition of 5 basic models of evolution. And then we'll add on a couple more additional parameters to those.